In this series of figures, we're going to take a closer look at the cochlea and its sensory organ, the organ of Corti. And so if we look at the cochlea up here in the upper left, just to back up a little bit, pressure waves in air would be converted into vibration of the tympanic membrane, shown by this oval I'm drawing here. Then we would have some auditory ossicles, the ear bones, that would connect the tympanic membrane through these auditory ossicles to the oval window of the cochlea. So what we're effectively doing is we're converting pressure waves in air to movement of the tympanic membrane, to movement of the auditory ossicles, to generation of pressure waves in the fluid of this vestibular duct to which this oval window is adjacent. And so if we unroll the cochlea, right, like just sort of schematically think about it and unroll it, right, we end up with this picture down below here, right, where we've got this sort of unrolled cochlea. Here's our stapes again, vibrating up against this oval window, which is flexible, and it generates pressure waves in the fluid called perilymph of this vestibular duct. Now, the vestibular duct, think about this is spiraling as we go up, spiral within the cochlea. But so as it spirals up, right, to the tip of that cochlea, known as the helicotrema, that vestibular duct is actually continuous with the with the tympanic duct. And so our pressure waves in the fluid here are going to eventually be transmitted over into this tympanic duct and be dissipated by the round window, another flexible window here at the base of the cochlea. Now, along the way, so where are we being sensitive to these frequencies of sound? Well, what happens is the reason for this spiral, the rationale for it, is that different frequencies of sound will pass across this middle duct, known as the cochlear duct, which is entirely membranous. They'll pass across at different locations. So high frequency sounds will pass across very early in the spiral, whereas low frequency sounds will pass across much later. Now, and what that does is it basically makes this cochlear duct and the organ of Corti within it an acoustic prism. And what we mean by that is it's separating the sounds by frequency to detect those frequencies separately. Now, our perception will be that those frequencies are detected simultaneously, but we actually detect them at specific spatial locations along this cochlear spiral. Now, if we were to take a cut through this cochlear spiral and then turn it sideways, we can sort of see those three spaces, right? We see the vestibular duct, right? Again, over here connected to the oval window and stapes. That, of course, would be connected to the tympanic duct down here, right? So those two are darker because they contain the same fluid, perilymph. And here's the cochlear duct in between with the organ of Corti boxed. Now, if we take another step, blow it up one more time, right? We're going to go over here, right? We're going to see a blow up of the organ of Corti. And so our pressure waves are going to be passing through, as we saw them over here, right? We see our pressure waves crossing over that cochlear duct across the organ of Corti. Here come our pressure waves. They're going to pass across this Reisner's membrane, right? That would be this upper membrane right here, okay? And then what you can't see in the lower magnification images is those pressure waves would cross through here and actually cause vibration of what's known as the basilar membrane. And that's found down here, right? And so the basilar membrane, right? So here's an arrow to it right here, right? And so this basilar membrane is vibrating. Well, the hair cells, which we've discussed before, are here and here, right? And those hair cells are being vibrated up against this tectorial membrane, this extracellular structure. And really its sole purpose is to create a barrier against which these hair cells and their stereocilia will be compressed, right? As we're vibrating this, the basilar membrane is bouncing up and down and it's squashing the hair cells, the hairs of the hair cells up against that. generating ion fluxes and output to the sensory neuron. Now, each location along the organ of Corti, along the spiral, is sensitive to different frequencies. So outputs from these hair cells are detected as a specific frequency, a specific pitch of sound. Now, why do we have inner hair cells and outer hair cells? Well, the inner hair cells are fairly easy to explain. Those are the ones that are receptive to these particular frequencies and sending signals to the auditory cortex to be processed, right? As loudness, as frequency, as other components of qualities of sound. The outer hair cells are essentially attached to the tectorial membrane. They're not sending a lot of sensory information back to the brain. What they're doing is they're being basically controlled through efferent output to regulate the degree of vibration or distortion that's occurring here. So they're sort of like a little bit of a buffer. And what they're thought to do is actually hone the frequency selectivity of the organ of Corti, basically aiding the function of the inner hair cells to detect specific frequencies.